proton NMR is extremely useful and it provides a lot of information, but sometimes there can be too much of a good thing. It can provide actually so much information that it can be really difficult to interpret. Proton NMR, when you have many hydrogen atoms, can be very difficult to interpret. So other nuclei can be more useful in providing information because there are fewer of them, and they provide simplified NMR spectra. And the two ones using in biological studies are carbon-13 and nitrogen-15. So this simplification, these simpler NMRs allows a more focused and specific approach because you are not overwhelmed by all the information of all the hydrogens that are part of that protein. For example, with nitrogen-15 and carbon-13 NMR, you can identify which specific amino acids might be involved in a particular process, in a particular reaction. So you can look at which areas of the structure are active or are responding to a particular ligand or that are involved in a particular process. You can study folding on conformational changes, oxidation states, reduction potentials of proteins, the effect of various physical parameters on the structure and the function, and if there is a mixture, the identification of the different components of the mixture, let's say a series of metabolites, and quantitatively, you can not only identify them but also measure them quantitatively, and also you can do ligand binding studies, whereabouts in your structure that ligand will attach. Carbon-13 and nitrogen-15 NMR for proteins requires isotopic enrichment, and that is because when you're looking at a proton NMR, you're looking at 99.98% of the hydrogens in your structure as being active in NMR. But when you're looking at carbon-13, a carbon NMR spectroscopy, only 1% of those carbons are actually carbon-13. And in the case of nitrogen-15, the most abundant isotope of nitrogen is nitrogen-14. You only have 0.3% of nitrogen-15 in your sample. So you do need to produce enriched proteins in nitrogen and carbon by, for example, by using recombinant expression. So if we look at, for example, this protein, the PDIA domain, in a normal expression, a normal PDIA domain would have 6 carbons in the whole structure. It would be carbon-13 and 1 nitrogen of the 159 nitrogens that are carbon-15. With a nice 90% isotopic enrichment, then you suddenly have 525 carbon-13 and 143 nitrogen-15. So most of your carbons and most of your nitrogens will be active in NMR, and you will be able to see the peaks and follow reactions in short periods of time. To the NMR is really useful in this field. For example, you can combine proton and nitrogen-15 HSQC, and it's the most popular, most widely used to the studies in protein NMR. HSQC stands for heteronuclear single quantum correlation, and it correlates a heteronucleus we saw before, for example, the example of carbon-13 in this case is nitrogen-15 with hydrogen, and it is one type of 2D NMR experiment. And the most common use for HSQC is to correlate the hydrogen to its directly bonded carbon or nitrogen, to identify to which particular carbon or which particular nitrogen that hydrogen is attached to. The hydrogen-nitrogen-15 bond correlations, the direct ones, are very useful because there's only one per peptide bond. So it is more targeted, it gives you a much more targeted NMR spectrum. So, for example, this is the proton NMR spectrum of one protein, and this would be which has, as you can see, many peaks, and this would be the HSQC proton nitrogen 15 NMR of that protein. As you notice, all the section between below 0 and 4 ppm has disappeared in the 2D spectrum, and that is because it's all the carbon aliphatic region. Those hydrogens are not attached to a nitrogen. However, the part of the spectrum that it is filled, it's the area where the hydrogens are attached to the nitrogens. Those are the hydrogens of the peptide bonds, and those appear in the spectrum. So suddenly, a spectrum that before was full of peaks, now it only has peaks in one section of the region. And notice that it's a sort of artifact provoked by the water peak. You have a line around 4 point something in the spectrum created by the water.